This is This Week in Science and Education, Episode 22, our fourth Q&A. It's time for This Week in Science and Education, a show that talks about science and health research and discovery and taking that information and bringing it into the classroom. This Week in Science and Education is brought to you by Mueller Systems. Need a backup? Got some important files you don't want to live without? Check out muellerdata.com/VROC. That's muellerdata.com/VROC. And by Polycom, world leaders in unified communication. Hey everybody, it's time for This Week in Science and Education. This is your host, Kevin Cougler, live from the University of Western Ontario. Joining me as always, let's go to Thomas first. Thomas Merritt up at Laurentian University. How are you, Thomas?
I'm well. Good to see you again.
It's always good to be talking to my favorite flute fly guy.
Great. Nice qualifier. Thank you.
Colin Jagoe from Kawartha is on the line as well. How are you, Colin?
I'm not Thomas, but I'm quite well, thanks.
We're off to a very ominous start. Everything's going wrong today.
We sure are.
We're going to pick it up right here, I promise.
Okay.
So our fourth Q&A, we've got a lot of questions to get to, but before we do that, I actually have a few housekeeping announcements to make. We've been talking for weeks now about a couple new shows that are coming up, so I want to make sure our audience knows about these. So the first one is called This Week in Engineering and Education, and much like T-WiSE or Science and Education, it's a show that will be talking about engineering careers and engineering research and with an educational twist, of course. That show will be airing next week, which is January the 26th, which is a Wednesday, and it will air every Wednesday at 3 o'clock p.m. You'll be able to watch that show live by accessing the VROC website at vroc.ca, or by going to the Ustream channel, which I believe will be ustream.tv/channel/This Week in Engineering and Education. But we'll have that link up on the VROC website for you.
Good call.
The next show, how am I doing? Am I recovering? The next show is This Week in Technology and Education, and that is also airing for the first time next week. Also available on the vroc.ca website, and that will air every Tuesday morning at 9:30 in the morning. So that's Tuesday, January the 25th, 9:30 in the morning. And the aim of This Week in Technology and Education will be to talk about software and hardware, anything technical related that's new or interesting, and again with an educational slant to it. So how do you take the new version of Microsoft Office and use it in the classroom? I don't know. I don't know what we're going to talk about, but it's going to be very, very interesting, I'll tell you that. All right, one last announcement. The forum is now active. For those of you that are teachers and students who'd like to post questions ahead of time, the way you do that is to go to the VROC website, vroc.ca, and look for the podcast forum. And prior to each show, you can log into the website and go to the particular episode. So, for instance, today's is episode 22, and you can, once you're logged into the website, post a question. Speaking of that, we have some questions here today, gentlemen, to ask both of you from our listening audience. So, shall we get right to it?
Absolutely.
All right. Well, this first one is a question about alcohols. And I just love the username. I'm not sure I can say this, but I'm going to say it anyways. What the heck? This is from Dan_smells. So, Dan, you might have a problem there. Maybe you should do something about that. But until then, here is the question. Dan writes, in a chemistry lab studying trends in alcohol properties, we studied ethanol, 1-propanol, and 1-butanol. It was discovered that the boiling points were 78.5 degrees Celsius, 97.1 Celsius, and 117.7 Celsius, respectively. I'm really anxious to see which one of you is going to answer this. Also, because the OH group in the cause of polarity, the longer the chain, the less polar the substance, as was discovered by testing the solubility in water and cyclohexane. Did I say that right, guys?
Sure did.
Sure.
Thank you for your support. Based upon these simple trends, I expected the melting points to follow a similar pattern, yet I found the melting points to be ethanol, minus 117 Celsius, 1 propanol, minus 126.5 Celsius, 1 butanol, minus 81.5 Celsius. This didn't make too much sense to me, as I expected either the 1 butanol to be lower than the 1 propanol, or perhaps the 1 propanol in between the two others, due to the decrease in polarity and increase in London forces. London forces? Okay. As the chain lengthens. Thomas, are you having a smoke? Oh, okay. A quick explanation of the reasons behind this would be appreciated. Thank you very much. That's from Dan Smells. So who would like to tackle that one?
It'd be appreciated and surprising.
It would be surprising. How's your background in chemistry, Thomas?
Yeah, that's right. I am in a chemistry department, and so I took advantage of that, and I emailed the real chemists. I have absolutely no idea what the answer to this question is. I do genetics. So I have actually, Colin, we'll see if you've got an answer for this, but I've emailed people that actually do chemistry in my department and said, hey, this is sort of cool. What do you think? So this may be one that we have to come back to next week with a quick answer from the chemists. What have you got, Colin?
Well, that's better than what I've got, actually. I could come up with a possible answer, but I actually did some digging on this, and I couldn't really. It is a very interesting question. I couldn't really find out an answer. I suspect the answer will have something to do with like the shape of it, the 3D shape, and the way once you get into a solid form, things sort of bond to each other, and the energy required to then melt and break some of those between molecule bonds might be different depending on the shape of it. But again, that's completely that's a guess on my part based on sort of what I did and some of the digging that I did. I'm gonna give Dan high points for a great question because I don't know. I'm not sure. And I actually talked to his teacher about it, and the reason they asked us is because she didn't know either. So it's a really good deep question that you try and figure out. Okay, well here's what I'm seeing, but why? Right, and maybe hopefully with Thomas's chemistry department there up at Laurentian, we can come up with a better answer, one that makes sense, and then we can get that posted onto the forum for Dan to have something better than just us two schmucks, pretending we know what we're talking about.
All right, so it sounds like that's a great question, Dan, and Thomas is on it. He's going to go his peers there and get the right answer. So we don't have it for you today, but it sounds like it'll be on the forum by next week. So check back, and thank you, Dan, for the question. Next question, completely unrelated. This is about animal evolution, and this is posted by Olima Muchest. Not sure if that's a first name and a last name, but or Olima Muchest. I'm not sure. At any rate, Olima's question is: if another animal on Earth was to evolve to near human intelligence, what animal would it be? Also, is there a chance that other animals are at our level of intelligence without our knowledge? I just love this, and of course immediately what pops to my mind is dolphins and porpoises, they say, are always very intelligent. So I'm really interested to hear what you guys have to say.
When I think about near human intelligence, I think about you.
Well, thanks for the kudos, but I'm not sure I even qualify there.
Oh, okay. Was that a little bit too much benefit of the doubt for you?
I'm editing that too.
Yeah. Okay. Well, we're still live, though. It's an interesting question, and it's one. I mean, I've heard this before. I think it was Stephen Jay Gould, evolutionary biologist, who wrote in some of his writing, if you wound the history of the Earth backwards and the history of evolution backwards, and then let it go again, you'd not get anywhere close to what we see today, right? It would be completely different. So to speculate as what animal might become, near human intelligence is really difficult, and it's virtually impossible to sort of make any really good guess at it, just because of the nature of evolution and the various factors that drive it and all of that stuff. But when I read the question, Kevin, I totally thought about, animal intelligence in general, and there's some really cool stuff out there about that, like you talked about dolphins and chimpanzees and various primates and monkeys, even pigs and dogs. There's a lot of whales have been recently found to be very, very intelligent based on some of the social interactions that they have. I was reading something about killer whales, and they're starting to really understand the social interactions between pods of whales, and there's thinking behind it. Like there's a lot of brain power of some sort. It's not like us, but it's definitely thinking. So it's very interesting. You know, in terms of what animal would it be? You know what? You can't answer that. Maybe Thomas has a better answer, but I'm not going to speculate.
Well, yeah, there isn't an answer to that. I mean, you can ask, what kind of intelligence do we find in the animal kingdom outside of humans, and even that's not an easy question to answer, because it comes down to what do you call intelligence?
Sure.
How do you define it? Yeah.
Yeah. How are you going to define intelligence? There was an interesting article in the New York Times yesterday about it's one of the herding dogs, and I'm blanking on the
border
breed.
collie or something like that.
It's a border collie, and this is a particular border collie that has this ridiculously broad vocabulary. And I think I've seen this dog written up before, but the ability of this one animal is causing us to rethink what animals are capable of, what they're thinking. Right? And we don't really have a good understanding of that. And Colin, I think that's where you're going with the idea of what whales are capable of, what's going on. It's unlikely that we're going to have sort of a Douglas Adams scenario where we're suddenly going to realize that the dolphins have been in charge all along, and they're going to thank us for the fish and disappear off into space.
And the mice are running the lab experiments on us.
Yeah.
Can you guys see Star Trek 4? It's not the dolphins; it's the humpback whales.
That's right. The one last humpback whale. I did see that Star Trek in the theater.
In 1972.
What's the next animal that's going to take over? There's no way to know. It's interesting. As an evolutionary biologist, one of the most difficult things to come up with are examples of evolution and process, which is really what this question is coming to. And we are getting better examples of that, and we've talked about some of the examples on the podcast before, where we can see changes in populations in real time. And that's sort of where this question is based on. So could we see differences in intelligence through time? I don't know of any examples that we have now where we have organisms that we can point to and say, this group is more intelligent now than it was at some period. We don't have an example of that we can point to. That general class of questions, can you give us examples of evolution in action, is a difficult one to answer, but we're coming up with better and better scenarios. Unfortunately, one of the best types of examples we're going to get of this is changes in response to climate change. And they're not going to be changes necessarily in intelligence, but we're going to be able to document changes in populations, in species through time. As we're drastically altering the environment that we're living in, we're going to see this evolution in action. One of the big issues here is the rate of time. Evolution we think of in evolutionary time, which generally means really long periods of time. But as we've accelerated climate change, and so our climate is now changing faster than it ever has in the past, we're going to see changes in evolution that are matching that. So I don't have an example for the intelligence question, but I'm afraid what I can tell you is keep reading the paper, and you're going to see lots of examples of evolution in action as we're drastically changing the environment.
And of course, the standard joke to other types of intelligence on Earth is we're not sure we have one type yet, given what we do to ourselves. So there's that too.
Yeah, well, that's a whole other show, I think, but that's definitely a great point, no question. Well, I'm thinking, gentlemen, we have 3 more questions that we want to ask, but maybe we'll just take a quick break now, and then we'll jump to these other questions. So before we get to them, I do want to mention our good friends over at Mueller Systems, who the company out of Woodstock, Ontario. They handle all things backup related. If you have some important documents and pictures that you need to get back up, I encourage you to log on to muellerdata.com/VROC and fill out the quick little online form. And once you do that, you'll be contacted and you'll be able to have access to the backup solution at Mueller's. It's wonderful. We do use it in-house at VROC for all of our backup needs, and we've had to restore before. And what can I say? The guys are just there if you need them. So we value our relationship with them, and their product is a good product. So muellerdata.com/VROC, and we thank them for their support. Moving on to some more questions. We have 3 questions here, fellows. I'll let you pick which one you want. The first one is heat absorption. We have a question on eczema, and we have a question on, what are these, operons?
Operons. Why don't we start with that one? That's sort of the easiest of the bunch to work on, and then we can work back into the less easy ones. So it says, why is it that only prokaryotes have operons? Why not eukaryotes? What control mechanisms are in place in eukaryotic DNA? So this is actually what I work on. This is one aspect of the research that we do in the lab. There's a trend so prokaryotics are simple organisms. They have a single or relatively simple genome. Eukaryotes are things that have a nucleus, mitochondria, that kind of complexity. Operons are strings of genes where you have a piece of DNA that codes for a protein flanked by a piece of DNA that codes for a protein and a piece of DNA that codes for a protein. All of these have a single regulatory element, and that's an operon. In eukaryotes, we don't really find that. Now, we find something similar where we'll have regulatory elements that are influencing more than one gene, but not this idea of an operon. And the operons are actually transcribed from DNA to RNA in one big piece, and then the protein is translated off of that. One of the biggest differences between prokaryotes and eukaryotes is not just the complexity of the organism, but the complexity of the genome. And with prokaryotes, they're a simple organism, a relatively simple genome, and the selective pressure seems to be to reproduce as quickly as possible. And reproduction means replicating that genome, making a new cell, replicating the genome, making a new cell. And so there's an enormous pressure for a very compact string of information and a quick turnover. That's where the operons come from. And because they're a relatively simple organism, then the control for the different proteins can be relatively simple. When you move into things like eukaryotes, the greater amount of complexity means that there's not, the operons aren't really as functional, or the idea of an operon isn't as functional in eukaryotes. You need more complicated regulation than an operon allows you. So if you have an operon with, say, 4 genes in it, all 4 of those genes are regulated in essentially exactly the same fashion. That's not entirely true, but it's largely true. In eukaryotes, you can't get away with that kind of simplicity. You need more complex gene regulation to support the complexity that's in eukaryotes. And so we, by and large, don't see something like the operons, at least not as prolific in eukaryotes as prokaryotes. So why not eukaryotes? Because eukaryotes are too complex. Why eukaryotes? Because they're relatively simple, and because there's this enormous pressure for a really complex, really dense regulatory region, coding region. So I've forgotten what the numbers are, but if you take the average gene in a eukaryote and an average gene in prokaryote, the size of the gene in eukaryotes will be between 10 and 100 times larger for a given gene in eukaryotes. That size isn't the protein. The protein size is about the same. That difference in size is all regulatory. It's all the pieces of DNA that determine how, when, where, how much for expression of that gene. So because of that greater complexity in eukaryotes, we don't see the relatively simple patterns of regulation that we see in prokaryotes. And that has to do with the fact that prokaryotes are relatively simple organisms. Eukaryotes are relatively complex organisms. So what control mechanisms do we see in eukaryotic DNA? The basic idea that promoters, the regulatory elements, enhancers are very similar between eukaryotes and prokaryotes, the individual components. It's just that there are a lot more of the components in eukaryotes than there are in prokaryotes. That greater complexity is because you've got more elements. It's like having a single switch versus having a switch, a dimmer, another switch, a backup, and then a switch that responds to something else. The pieces of the switch are relatively similar between the two. It's the number of switches that we find in eukaryotes versus the number of switches that we find in prokaryotes. So there you go.
Okay. That's great. I have a question, though.
You're supposed to give the answers.
No. It's made me think of something else, and so I'm going to ask a question. I've got Dr. Thomas Merritt on the line. I'm going to ask him a question. That's my prerogative. In an operon, in a prokaryote, so you've got one regulatory section, and then say there's 4 proteins that can be coded for. At what point do those proteins then become distinct proteins? Are they all made at once? Are they made a bit at a time? How does the RNA then translate that into proteins? At what point do they become separate?
Yeah, so now we're pushing past what I actually do into what I remember from when I taught this.
Okay,
the RNA comes off as one long strip. The ribosomes take the RNA and translate it into proteins. So the proteins are actually translated as individual units off of that one message. So you have one string of RNA, but that RNA is being translated at different points to make different proteins. So the proteins are always created as separate entities, 1, 2, 3, 4. The message is one long string, and where the ribosomes bind the message determines where that protein starts.
Right. Okay, so there's they're making them all at once off the same strip of RNA then.
That's right. So the RNA is just the information, and they're actually the ribosomes are taking
which is why it's so fast then to make multiple proteins at the same time, because it all makes sense now. Well, good. Well explained.
And so when you look at this, you find that all of those proteins are working in the same function, right? So they're in the single pathway, and so protein 1, 2, 3, and 4 are at different points in this one pathway. They're all needed under the same conditions, and that's where this comes from. Now, in eukaryotes, there are often times where we always need these 4 proteins for one pathway, right? So why not an operon? Well, because instead of having an operon where they're all controlled by a single element, we've got more complexity where we've got multiple elements controlling them individually, and that gives you more flexibility on where those components can go. And so now we can have components 1, 2, 3, 4 present for this pathway, but 1, 3, 5, and 7 can be present for this pathway. So the lack of operons gives us more complexity, or the ability to code for more complexity, but it means a much larger piece of DNA, and in prokaryotes, smaller is better, more or less. Kinda.
Cool.
That question came to us from a student whose username is HLSS4B3. So we have no idea who that person actually is in real life, but I think that HLS stands for Heart Lake Secondary School, so I believe that's out of the Peel region. So wherever you are, HLSS4B3, we thank you for that question. That stimulated some dialogue, and that's what the show is all about, one of the fun things that we do. So thank you very much. Just to cap that off, Colin, what grade level are we talking about with that type of a question, do you think? Definitely secondary school, I imagine. Are we in the higher grades here?
Yeah, that'll be, grade 11, 12, talking about, genetics and protein creation and, translation of DNA into RNA and into proteins. So I'd have to dig out my curriculum and look out specifically which one, but grade 11 and 12 biology, somewhere in there.
Let me throw an interesting twist into that, just for the idea of, when you're teaching this to your students. So there's an operon called the LAC, L-A-C operon, and this is one of the models for genetic regulation. And if you talk about any operon in high school, you almost certainly talk about the LAC operon.
Yeah.
And LAC, it stands for lactose. And so this is a group of proteins that are involved in lactose metabolism. And this set of genes that are all in this chunk of information were named for the fact that they're involved with lactose metabolism. It ends up they are, but the proteins, this operon, is found in organisms that have nothing to do with lactose. Lactose is milk sugar. And from a human perspective, we often thought of the lactose operon because it's the group of genes that are in bacteria that are involved with digesting milk sugar in the human gut. They're found in E. coli and in other organisms that consume milk, so cows and other mammals that are producing milk, right? So it's this very mammal-centric naming of the group of genes. The LAC operon are the genes that are involved in lactose metabolism. That operon is a very, very ancient operon and is found in many bacteria that have absolutely nothing to do with lactose. And so here's this model that we use for gene regulation, named for the protein that this set of genes evolved to metabolize lactose, except they didn't evolve to metabolize that. They evolved to metabolize a group of sugars that includes lactose. But we named it because to us, these sugars are lactose. This is milk sugar. As far as the bacteria are concerned, from an evolutionary perspective, this was a group of genes that are involved, that have evolved in bacteria for sugar metabolism, nothing to do with lactose. There was a student when I was a postdoc at Stony Brook that was actually looking at how widespread this operon was, and one of the interesting things he found was that it is very widespread, and it has nothing, by and large, it has nothing to do with lactose. It's just our own perspective that to us it's important because lactose is important to us as a species.
Right. It's all about us.
Okay. Thanks, Thomas, for that. Now, how are either you or Colin with your dermatology?
So I looked this up.
Yeah, you have something we need to look at, Kevin? You're not going to show us your rash or anything, are you?
Yeah, listen.
Another editing opportunity. Thanks a lot.
You don't pay me enough to show me your rash.
All right, at the risk of getting too out of control, I'm going to read this question. So this comes, again, I think, from a student at Heart Lake Secondary School whose username is HLSS4M21. So HLSS4M21, if you're out there, thank you for this question. This student writes, can having eczema cause your skin pigments to change color? If so, does it matter if it occurs at a later age? Thanks for that question. Well, who's the bravest? Who wants to tackle that one?
So I looked into this a little bit, and it ends up that eczema is similar to other diseases that we've talked about in this podcast, where the example that I always give is cancer, right? And so cancer is this umbrella term that refers to hundreds or more likely thousands of different diseases that all have this shared feature of uncontrolled cell division. So eczema is a blanket term, an umbrella term for a series of skin conditions that have a similar rash. Within that, there are a number of different types, some of which are hereditary and some of which are not. And with the little baby that we have in the house, my wife had eczema when she was an infant. And there are all these stories about having to keep mittens on her so she wouldn't scratch at the rash, all this stuff. So we've been watching little Lila for the last 8 months to see if this is going to crop up, because there is this hereditary component. So the first thing you think of when you see a hereditary component is genetics. So is there a genetic component to eczema? And the answer is yes and no. So of the dozens or maybe hundreds of diseases that are classed as eczema, some of them are hereditary and some of them are not. All of them seem to have this sort of common feature of possibly a response to the environment. So even the hereditary forms of eczema are not always manifest, and they seem to crop up if there's something else. So you've got this now an interplay between environment and genetics. And as a geneticist, this is always exciting, aside from the itchy, rashy part, where you're looking for this interplay of both the environment and the genetics. And this is classic for disease, that neither one of these act in isolation. So what triggers eczema? Is it a genetic component? In some cases, absolutely. Is there an environmental component? In some cases, absolutely, and it seems to be a combination of those two. Now, does it have anything to do with skin pigmentation? Eczema, and many other skin conditions, can lead to scarring. Scarring will change patterns of pigmentation. And so, I'm not this kind of doctor, but my impression looking through this is that if you get changes in patterns of pigmentation, it's more likely that this is a secondary cause through an infection or a scarring than from the rash itself. Does it matter if it occurs later in age? It ends up that some of the apparently hereditary eczemas are early and some of them are later. And so I doubt that the pigmentation has anything to do with whether it's hereditary or not, or whether it's early or whether it's late. But that what we're looking at is a secondary reaction to the actual the scratching that comes in to the first disease in the first place.
Yeah, and when I looked into it, that's exactly what I found, that it in itself doesn't seem to change any pigments, but when you get scarring, when you get other like you said, secondary effects, it can lead to, things like that could eventually, if you get a scar from anything, it's going to change the skin color because that's what a scar is, right? But yeah, that's sort of what I had figured out as well.
One of the interesting things that I saw reading through this briefly was that while eczema does seem to be associated with things like asthma, other sorts of allergic reactions, it's not certain exactly how that association works, and it doesn't seem to be a simple association. So higher incidence of asthma and other immune function diseases do seem to be correlated with higher rates of eczema, but it's not a perfect correlation. And so having one or the other doesn't mean that you'll necessarily get the other one. But there does seem to be this correlation, and this may come back to the environmental component, that the environmental issues that are promoting things like asthma may also be promoting things like eczema. But we really don't know enough about it to know exactly how that's working. But I think that's the direction that people are taking some parts of the research.
Okay. Well, thanks, guys, for those answers. And thank you again to username HLSS4M21, whoever you are. We appreciate that, that question. You know, guys, I have to tell you that, you know, I really enjoy interviewing the different guests that we have on the show, and I enjoy going to the Snow Lab and going on location at Science North, those types of things. But these question and answer shows that we do, I really find them interesting, and I have to say these are, you know, some of my favorite shows just because you guys are such brainiacs. I feel comforted knowing that people like you exist and have answers to some of these questions. It's absolutely fantastic. So, listeners out there, these guys are serving you well. With that, I'm segueing into the last question of the day, which we may not have an answer to.
We got this one covered.
I was figuring that I've had so much to say about the previous questions that rather than steal Colin's thunder, I'd let him take this one.
I appreciate that, Thomas. Thank you, sir.
I'm here for you. I have no idea what the answer to that question is. Let me be honest. Seriously, no idea. I have no idea. None.
So I'm going to Jagoe on this one, is what you're saying.
Yep.
All right, so Colin, here you go. This is directed at you.
Shoot, read it out.
And this the topic is how color affects heat absorption. And this was asked by Gene or Genie1. It's a physics question. Here's the question. Actually, I'm going to personalize this. Dear Colin, I am doing an experiment with color and light.
Awesome.
He personalized it, but then he left. He dropped off.
Dear Colin, I never
shall we continue without him?
Shall we assume? Yeah, just answer the question.
Let's just keep going. Absolutely, he can edit himself back in later. My topic is how color affects heat absorption. How do the waves absorbed by a color produce thermal energy? Why do the longer wavelengths produce more energy? I want to explain why cooler colors, green, blue, and violet, have a higher temperature when placed in front of white light than warmer colors, red, orange, and yellow. Basically, how does thermal energy relate to the absorption of specific wavelengths and colors? There's actually a couple of things with this one. In your question, Gene, you ask why cooler colors, green, blue, and violet, have a higher temperature when placed than warmer colors. Calling things warmer and cooler colors is just the way we do it. It doesn't actually have anything to do with the energy inherent in the light itself. So, just because we call red sort of warm colors and blue cooler colors has nothing to do with the actual energy in the light itself. When you actually look at these things, and the easiest way to remember it, always in my mind, is sort of the difference between infrared light, which is infrared radiation, which is heat, which is longer wavelength. It's close to red on the end of the light spectrum. If you go past red, you got infrared. And then you've got, on the other end, blue and violet, and then you get into ultraviolet, right? And ultraviolet is very high energy radiation. It's the kind of radiation and light, quote-unquote light, that you can get sunburns from, and skin cancer, and melanomas, and all these other things. It's very high energy. So the blue light, right, off into the ultraviolet, actually has a shorter wavelength than the red light does on the other end, which is close to infrared and heat energy. So when you actually look at the wavelengths of light, red light has a longer wavelength, blue light has a shorter wavelength. Short wavelength equals higher energy. So blue light has more energy inherent in it than, say, a red one does. And that's you got to keep in mind the difference between heat and infrared, though, too, right? Because molecules react differently to different wavelengths. We can feel infrared on our skin, but we can't necessarily feel ultraviolet. But heat is only one kind of energy, so you're talking about thermal energy here. When you look at then how it absorbs and re-emits it, there's a couple of things you need to think about. And you're probably familiar with, sort of the black versus white. If you have a black car versus a white car, it heats up the black car because that energy is all absorbed because nothing is reflected. And then a white car reflects a lot of energy, so it doesn't heat up in the sun as much. With specific colors, right, you have to look at each color specifically. So if you've got something that's shining with a blue light, and in your thing here you talk about explain why cooler colors, you say blue, green, violet, have a higher temperature when placed in front of white light than warmer, right? And the reason is because the blue color is actually higher energy. So if you have something that's blue, it's absorbing a higher energy wavelength from the white light, which has all of the colors, and then that energy itself is turning into heat and re-radiating, and that's the temperature that you're measuring, if that's in fact what your experiment was. So because blue
Colin?
is absorbing more energy, yeah.
I may be remembering this incorrectly, but if you see something as blue, isn't that what's actually being reflected from it? So it's absorbing everything
That's
except
true.
blue.
Yeah, that's true. That's true. It's absorbing
So, I
everything
mean,
except blue.
not to point out that everything you've said is actually completely the opposite of what we
backwards,
see, but
backwards, yeah. Well, see,
Hey, I'm sort
again,
of back. Can you guys see me?
Yeah, we can
What?
see you. I guess the question, I'm not sure how the experiment is being run. Like, are they using
I
blue
think they've
light,
got a piece of...
or are they using something blue that then they're measuring the temperature of?
Yeah, I think what they've got is white light and then something in front of it that's blue, right? And
I would
so,
suspect. Like a
so
blue filter,
if-
maybe.
Well, now, see, there's a question. I don't know.
Right?
Because if it's reflecting blue, then it's absorbing everything except blue. And if it's, et cetera.
And if it's a blue filter, then the blue light is coming through, which would then be higher energy than, say, coming through a red filter.
But it's absorbing whatever's not being filtered. This is why we need a This Week in Physics,
Yeah.
which would be even more exciting than This Week in Engineering. Let me tell you, I will be setting my clock so that I can listen to This Week in Engineering.
You think we need to bring this one up, eh? This Week in Physics.
But
All right.
now you've got me-
So we're
now
gonna
you've
have
got me
rivalry
thinking
now
about it.
between the shows. Oh, great. Well,
Cool.
I think-
were you able to have some type of an answer to this while I was gone?
We did, but now we think it's
We
wrong.
were, but now I'm not sure. I'm not sure how the experiment is being run, to tell you the truth, right? Where I was going with that was just that, blue light is higher energy and red light is lower energy, and if something's absorbing, and you're right, Thomas, if something's absorbing blue light, it theoretically should be then heating up more simply because of the energy, it's getting more energy. But if it's blue that you see, this is where optics and color gets complicated, so it's reflecting blue light and absorbing everything else. It also could simply be to do with like the darkness and lightness of the material as well.
Well,
Right.
the reflectivity.
The
So the
reflectivity
more
and the albedo of the material, right? So there's a lot of things I don't know here about this particular experiment.
That's what I wondered, and not actually
Yeah.
working in physics, and it's been a long time since I took a physics lab. But if what's determining the difference in heat is how much light's reflected versus how much light is absorbed, so I'm not sure.
And
what the
that
answer to
would be
is.
correct, right? Like, yeah, that absorbed versus reflected is why things heat up, just in general, with white light, right?
Well, fellas, I'm going to suggest something.
Kevin, over to you.
How about we offer to throw this one into the Ask the Expert forum, and we'll get one of these physics people to answer that for Jeannie. What do you think?
Mm-hmm.
Awesome.
I think it's a great idea. I mean, it's a very interesting question. I'd like to know more about her experiment, exactly how she was running it. Was it filters? Was it material? And then maybe we could dig into it. But on its own, maybe somebody trained in physics would have a better answer than either Thomas or I.
Right. So there you go, Jeannie. Thanks very much for the question. We do appreciate it. The guys took a stab at it, and we'll throw it into the forum and get a physics expert to follow up with that question. So, well, gentlemen, I think that's another show for today. Any last thoughts before we sign off?
Oh, let's end on a low note of having no idea what the answer is.
Well, we can only improve on the next Q&A, so tune in next month for the Q&A, and we'll see what we can do then. So, well, gentlemen, on behalf of Thomas Merritt at Laurentian University and Colin Jagoe at the Kawartha Pine Ridge District School Board, this is Kevin Cougler, and This Week in Science and Education. Thanks very much for tuning in. We'll see you all again next week. Have a good week, everyone.
Thanks, folks.